So we are ready to start with our last session of the day. The session title is Disease Management Strategies, and it will be presented by Dr. Jan Grusset. I tried my best.
Okay, thank you. Welcome to this session. We'll have 4 presentations today by at least 3 very distinguished presenters. The 4th one I can't have an objective opinion about because it's me, so I'll let you decide. So the first one is going to be a pre-recorded presentation, but by a very distinguished scientist indeed, Dr. Sophien Kamoun. So Sophien is a group leader at the Sainsbury Laboratory and professor of biology at the University of East Anglia in Norwich in the UK. And his research focuses on using genomics and molecular biology methods to understand plants and filamentous pathogen interactions, from pathogen effector biology to plant immune responses. Sophien studied genetics in Paris and Davis, California, before working in Wageningen, where I met him actually the first time, Ohio and Norwich, where he kind of stayed, and he's based there now. And he served as a head of Sainsbury Laboratory between 2009 and 2014. And he has received numerous awards and honors for his outstanding contributions to plant sciences. In 2018, he received the Linnean Medal and became the Fellow of the Royal Society. And Sophien's research has resulted in cutting-edge methods to mitigate some of the world's most serious crop diseases. And I can attest to that. He worked a lot on late blight, and several of the genes that he identified we've been using in our breeding programs as well, but also in transgenics. But maybe a more interesting story is probably if it wasn't for Sophien, I wouldn't be here today. Because back in 1995, there was a lady doing an Erasmus exchange in his laboratory where I met her. And she now is my wife. And that led to a whole series of events that eventually led me to Latin America and Cabana, and now I'm here. But anyway, that's maybe with that story, you'll remember Sophien even better. So I'll let you start the presentation.
Hello, everybody. Thank you for inviting me to join this event. And I'm sorry that I could not be there in person. What I would like to do today is tell you about plant health and plant health emergencies and how we can address them with the science of genomics. So I'll start with this slide taken from a nice article written by my colleagues Matt Fisher and Sarah Gurr a few years ago, which highlights the increase in frequency of fungal diseases throughout the world. And this is particularly acute in the case of plant pathogenic fungi, as you can see in that graph. The reasons, of course, we all know them: increased global trade, climate change, pathogen host jumps, and perhaps even more recently, armed conflict is now an issue, given that the Ukraine situation is resulting in shakeup in the trade of agricultural commodities and most likely an increased risk in new introductions of plant pathogens. Of course, this is only about fungi, but the same would apply to other types of plant pathogens. And as you know, many plants are infected by a variety of pathogens, not just fungi, but also viruses, bacteria, fungus-like organisms, and also a number of pests like nematodes and insects. But this slide is what saddens me. If you go to the ProMed Mail service and look up plant diseases and plant outbreaks, you'll see that the majority of them are still undiagnosed. And this is really sad in this day and age, because we have the technology to rapidly apply genome science, DNA sequencing to diagnose a variety of these pathogens. And if you do word cloud of all the text that's present in the plant section of the ProMed mail server, you'll find out that the word undiagnosed is the most common. And most often, this is in developing countries. And the identity of the pathogen is not known. And in many cases, we don't just want to know the identity of the pathogen, but we want to know even what type of race of pathogen is out there causing the outbreak. So I found this very saddening, and about 10 years ago, in fact, precisely 10 years ago, I was inspired to write a commentary on how we should apply very rapidly, without waiting, genome sequencing to any emerging plant disease, in the same way that it happened more recently with the SARS-CoV-2 and with the COVID pandemic. We really need to react very quickly and not wait for doing the classical plant pathology work before we do the genome sequencing. And as you know, this is routinely done with human pathogens, and I suspect you have discussed this at this meeting. So we can't afford to wait. We really have to apply sequencing as soon as we can, as soon as there is a new outbreak that's detected, because the information generated from those sequences would be very valuable. And the talk today will illustrate the value of doing this type of analysis and doing it rapidly. Since 2012, there was some development. For instance, there was an outstanding workshop that took place at Bellagio, Italy, where a group of people led by Monica Carvajal from CIAT in Colombia. And Monica led this group to discuss how we could implement a global surveillance system for crop diseases. And one component of that discussion involved genomic surveillance. And the conclusion was quite evident, I think, to many of us. We need more data, but we need a lot more data. We need 1,000 times more data. Speed is critical. And we should sequence also directly from the field. We don't need to necessarily wait until the pathogen is cultured. And in many cases, we have obligate pathogens. We cannot even culture these microbes. We also need better integration and better networking between the genomics people and those collecting other types of data. That data is very valuable in interpreting the genomics. And finally, and this is also a topic I want to preach upon today, is the importance of open science and also the importance of crowdsourcing and the importance of reaching out to the community as a whole to find volunteers who are willing to contribute to solve the problem. So essentially what we need is to rapidly and effectively respond to this emergency. What we need is a new way of doing business, a completely new way of doing business. We need to do away with the model of hierarchical networks and hierarchical collaborations where there's someone on top, maybe an international agency, that is directing people on what they should do. We need to move to a more open system where anyone who's an expert can step in and contribute their bit. This is messy, it's true, but it's more likely to result in the optimal solution and the most rapid response to the problem, because you are automatically recruiting people who are self-motivated to help and also who are the best expert to address the problem. So let me tell you what happened with the wheat blast outbreak. This is a story that will illustrate the points I want to make, so that's why I'm going to cover it. Some of you must have heard this because it's already dating back to 6 years ago, to February 2016, when an outbreak of a wheat disease popped up in Bangladesh. They never seen any disease like that, and it did look like it was caused by the blast fungus, Magnaporthy oryzae or Pericularia oryzae, depending on the authors. This disease was very destructive. As you can see here, there's really thousands of hectares that were destroyed. And often this was 100% yield losses. And also because the disease is seed transmitted, it pushed farmers to burn their fields. In fact, there was even mandates to burn fields, affected fields, to avoid the seeds being kept and replanted again the following year. Very dramatic situation. This story dates back to 6 years ago, but you'll see that there is a new twist in this outbreak or in this disease, and I'll cover that later on. So what happened back in 2016 is I was in communication with a friend of mine, a plant pathologist of Azal Islam, based in Dhaka in Bangladesh, and we communicated through Facebook about this outbreak, and we indicated to Tofazal that we have the capacity to sequence samples directly from the field without waiting for culturing and so on. And so Tofazal sprang into action. He's a very dynamic fellow, and he contacted many people using Facebook, as a matter of fact. So social media is actually part of this story. And he reached out to many extension specialists and farmers throughout the affected districts. And we got many samples out of that very, very quickly. The way we decided to approach this is to take all the data we have, and as soon as we had it cleaned up and QC'd, we posted it on a portal we called Open Wheat Blast. At that time, I never worked with Wheat Blast before. I had little experience in this disease, in this pathosystem. And so I really, personally, my lab couldn't really do much. We had to rely on experts coming in. Essentially, we served as brokers between Tofazal and the community. And we decided to reach out with media and with an open data set to reach out to collaborators. Anybody in the world who wanted to collaborate could collaborate. So we did that, and this shows you the timeline. So within essentially 6 weeks from field collection, we had the first report posted by the community telling us essentially the answer to the question of where did this pathogen came from. So field collection occurred on March 16, 2016. We received the samples, performed the RNA extractions, library constructions, and all of that around Easter 2016. Open Wheat Blast went live on April 18th. And then the first report, and there was at the end 2 major reports that were submitted. The first one came from Daniel Kroll in Switzerland, at the time in Zurich, currently in Neuchâtel. And he posted the first report on April 27th. So that was 6 weeks after feed collection. Pierre Gladieux, also from Montpellier, soon after that, posted also a report reaching the same conclusion. So the conclusion was that the population analysis based on the sequences from the field indicated that the wheat blast in Bangladesh was of South American stock. So most likely it was introduced to the country through exports of wheats, possibly commercial exports. But we know that at least one South American country, Brazil, exports wheat to Bangladesh. So that's perhaps one route through which this pathogen moved to Asia, to Southeast Asia. I just wanted to highlight here bioarchive. So again, March 16th is when the samples were collected, and June 19th, so 3 months after field collection, we posted a paper that synthesized the reports we received through the crowdfunding. So this paper was a community exercise, but many people contributed data. In fact, when we went public, there was only one single genome of a wheat blast isolate that was available, publicly available, but turned out that the community had many other data sets. And at the end, when we went live, we had 22 genomes that were part of this study. They were sitting on people's servers, essentially, waiting to be published one day, but they were very valuable. Without those data sets, since all of them were South American, without those data sets, we wouldn't have been able to interpret the origin of the Asian outbreak. So 3 months later, we posted it in the preprint server by archive, and 3 months after that, in September, we formally published the paper, as you can see here. So the timeline was really quick and rapid, and I think this is an exemplar for how we should respond to plant health emergencies, and not just in terms of sharing the data, but also producing these community papers where everybody is getting credit for the work they did and the contributions they did. Okay, just a little bit of science here, because I think it's a little interesting how we did it. We applied a method we call field pathogenomics, which was pioneered by Diane Saunders, currently at the John Innes Center. And what Diane does is she basically does RNA-seq, so sequence the transcriptome from lesions, both symptomatic and symptomatic tissue from the affected area. And using some bioinformatics tricks, you could separate the pathogen sequences, or let's say most of the pathogen sequences, from most of the host sequences. And as you can see here, that cloud of blue are actually fungal transcript. And even in cases where we have very little fungus infection, the scatter plot on the left is 0.5% of the reads, only 0.5% aligned to the fungus. We still can identify not just the pathogen, but also some cases, the lineage, or maybe even the race of the pathogen that's present in that field. So this is the advantage of doing RNA-seq. It's very quick. And Diane in her lab, she applies this to the rust fungus, yellow rusts, and the rust fungus cannot be cultured. So if you want to really get the genome sequence, you have to invest several weeks, if not months, of trying to establish a culture in the lab on wheat plants before you can generate enough material to sequence the genome. So this method is very useful. Of course, once you have isolates of the fungus, you can sequence the whole genome, but as a quick scan of what's out there in the field, it's a very appropriate method that I encourage you to consider. Again, more data. This is simply showing that you can see there on the right that these are all the green samples are wheat isolates from South America, at the time primarily Brazil, only Brazil, and then the Bangladeshi stock, if you like, the lineage, is nestled within these Brazilian isolates. So the conclusion is that we have a South American introduction from to Bangladesh, to Southeast Asia. Now, fast forward to September 2020, and a paper came out, this paper, reporting that wheat blast was now detected in Zambia. This paper didn't have a lot of genomics, but it had something that really worried me and to some degree upset me a little bit, because even though the paper came out in September 2020, the outbreaks dated back to 2017 and 2018. So for some reason, even though this is a team of international scientists, they knew that the pathogen and that the disease was in Zambia back in 2017, but the news only reached many of us in September 2020, so about 3 years after the initial outbreak. Not only that, the paper was actually under review for about a year, apparently, based on what the journal reported. So they could have posted the paper as a preprint a year earlier, and they would have again alerted the community to this outbreak, to this plant health emergency a year earlier. And this is no joke. This is a completely new pathogen popping up in a country where wheat is actually important in the southern part of Africa. And of course, in North Africa, it's not just important, it's actually absolutely vital for the diet of North Africans, which is where they get 50% of their calories from wheat. So there's reason to be concerned, but what I really want to illustrate, I don't want to really blame the scientists here, but what I want to illustrate is the fact that the culture of our scientific culture, our academic culture, is just not up to date in dealing with these kind of outbreaks. So finally, after the paper came out, we were in touch with colleagues, and particularly Alison Bentley, who took over as head of wheat at CIMMYT with very progressive ideas and with the wish to modernize their approaches and embrace approaches like open science. And so we obtained isolates from Africa, from Zambia, and we wanted again to go quick. In this case, we had isolates, so we could have sequenced the genomes, but we could go even faster by doing multiplex amplicon sequencing. And this is a set of primers, about 76 initially, but then at the end added up to 84 markers, so signal nucleotide polymorphisms, flanked by primers that we could very quickly do the PCR, multiplex sequence them, and determine whether this Zambian outbreak was caused by the same stock that caused the Bangladeshi outbreak or not. That's how these SNPs were designed, really, to follow whether in Bangladesh we have new incursions and whether this particular lineage would pop up elsewhere in the world. So that was very fast, and I should mention Kurt Lamour here at the University of Tennessee, who developed the monsterplex method. It's a multiplex amplicon sequencing approach. And what Kurt does that through his company, Floodlight Genomics. So just to inform you that this is a commercial service. But it's really beautiful technology because you can sample hundreds of samples for very cheap and get a lot of information very quickly. So the science was that, as you can see there on the left side, the Bangladeshi and Zambian isolates were identical in all 84 markers, just identical. There was not a single polymorphism in those 84 markers, even though we compared a data set of over 500 Magnapothi oryzae blast fungus samples from all over the world, as you can see in that map, and also through different periods of time. So it does look like we have a clonal lineage, most likely, that somehow reached both Southeast Asia and Africa. And amazingly, it's the same lineage. We went on to do genome sequencing to get better resolution, and then we could identify sublineages within this pandemic clone. And you can see here that the Zambian and Bangladeshi sublineages are actually split, and the closest isolate from South America to this lineage is a strain called B71. It's actually from Bolivia, and it is related, of course, to the Brazilian populations. So the B71, we like to now refer to this as the B71 clonal lineage, because B71 is the oldest isolate we have from this clone. Apparently, out of that clonal population in South America, there was 2 introductions, one to Bangladesh and a separate one to Zambia. And these clones are very recent, as you can see here. The PopGen, the population genetics, the population genomics analysis reveals that they're about 10-year-old at the most. I should highlight that this work was done by primarily by Sergio Lator and Hernan Burbano, so 2 Colombian expats who are based at UCL, and they are fantastic PopGen collaborators. Check their lab; they're really interesting, doing really interesting work on BLAST and other systems. Okay, so the next question is: okay, it's fun to do all these genomics and determine the origin, but can genome analysis really inform disease management strategies and maybe suggest some solution to these problems? So we did a couple of things in that sense. The first one was to scan these genomes, the pandemic genome, for effectors, so the secreted proteins that are targeted by the wheat resistance genes. And we found that one gene called AVR-RMG8, this AVRMG8 by chance, by pure luck, happened to be present in the B71 clonal lineage. It's invariant in both B71 and the Bangladeshi and Zambian populations. And also it's associated with other factors that make us predict that this lineage cannot infect weeds that carry the RMG8 R gene or resistance gene. So plants have resistance genes and they can fight off pathogens, and RMG8 is one of those genes that's present in weeds. But the point is that, as you can see on this cartoon, that the majority of the South American population don't carry the AVRMG8 effector, just because this is a variable, highly variable protein, just like the spike protein in SARS-CoV-2. But through the luck of the draw, the pandemic clone happened to carry the allele of the effector that is detected by the R gene. So we went on and tested this prediction. I should mention that this was done by Vincent Weir in Nicholas Talbot's lab at the Sainsbury lab. And essentially we confirmed that indeed wheat plants that carry the RMG8 gene cannot be infected by this pandemic lineage. And that's really what this slide shows, that if you have RMG8 in your wheat plants, then the wheat plant is doing quite well with all the isolates we tested from Zambia and Bangladesh. So that's good news. So here's a case where the genome analysis very quickly point us to an R gene that can be now used to manage this disease. The second question is fungicides. We know that fungicide resistance in the wheat blast fungus is very common in South America. There's a lot of spraying going on, and therefore the fungus has evolved to become resistant. And we also know that the popular strobilurin fungicides that are used quite a lot against the blast fungus, we know the mutation that results in the fungicide resistance, in strobilurin resistance. It's a mutation in the mitochondrial DNA. It's a single amino acid. I don't know if you can see it there on the lower left. It's this G143A polymorphism in the cytochrome B gene that results in fungicide resistance. So the purple samples are the ones that carry the fungicide resistance. And you can see that essentially the majority of the South American isolates are resistant, are predicted to be resistant to the fungicide. But B71 and the pandemic clone are not. They don't carry this resistant mutation. So we predict from the genome analysis that strobilurin fungicide will work against this pandemic clone. And we tested that, and indeed it does work. So here you can see sensitivity to azoxystrobin, which is one of these fungicides. And indeed, all the isolates we tested were sensitive to the fungicide. So that's another good news. Through the luck of the draw, it happened that the pandemic clone was sensitive to fungicides. However, there is an if here. We know that the fungus can evolve to become fungicide insensitive or fungicide resistant. And we did the test in the lab. We wanted to test the evolutionary potential of the fungus. Can it evolve to become resistant fairly quickly? And indeed, we did manage to make fairly easily a variant of this pandemic clone using a Zambian isolate. This is the one labeled SR1. It's present twice on that panel on the right, on that petri dish on the right. And SR1 became resistant to azoxystrobin, and it also carried that exact mutation I described to you earlier. So understanding also the evolutionary potential of the fungus can also help guide disease management strategies, maybe fungicide treatments, and so on. Knowing that this can happen is quite important. Again, with the question of the evolutionary potential, there is actually potential disaster that could happen out in Africa. And this illustrates also the kind of indirect problems that could arise, is that in Africa, we have a wild population of Magna porthalis, if you like, or Magna porthiorizi, and other related pericularies. There are actually some endemic African lineages and populations. For example, the finger millet, this is an isolate or a population that's well studied in Africa. It causes actually a nasty disease on finger millet, which is a crop. And that is widely present in Africa. So we tested whether the clonal lineage and whether strains isolates belonging to that clonal lineage are fertile with endemic African strains, and indeed they are. There are strains that belong to different mating type in Africa, and that can mate and cross with the newly introduced wheat blast fungus. So as you can imagine, this would be an experiment I wouldn't want to do, to basically cross these two and generate a progeny that could potentially yield an even more aggressive or more virulent form of the blast fungus, whether on wheat or on other crops. I should highlight that this and the previous work was done in Nick Talbot's lab by Andy Foster and Vincent Weir, two members of our network and our collaboration on this project. So that's the story, and I think there are many lessons here to take. One is that genomic surveillance can be quickly applied to monitor plant disease outbreaks and pandemics, and we should really do this at a larger scale. And I think we need coordination, for example, through the global surveillance system, also some sort of international network that allows us to rapidly apply genomics to new diseases. I hope I showed you that valuable information about the identity and origin of the pandemic agent can be gleaned from sequences and fairly quickly. And also genomics can reveal and guide maybe some disease management strategies, for example, which R genes could be used, or the status in terms of using certain fungicides. But again, here we can also do lab experiments to highlight the potential, the evolutionary potential to have new variants. And I wish to see this concept of variant of concern that's now a popular concept given the COVID-19 pandemic. I think variant of concern is something we really need to integrate also in plant pathology, and a very nice, I think, reason why we should be doing more genomic surveillance and more sequencing of these various outbreaks, just to keep an eye on them also. And finally, the approach of crowdsourcing, this really helped to bring experts and the community together. And what was really nice is we had experts really coming out of the woods, if you like, and contributing their analysis at various levels. Even with the lab work, we actually had people stepping in and saying, hey, I'm experienced in doing these fungicide assays, or and so on. And so this leads me to the last part of my talk, which is that we really need to rethink how we approach the way we do our science during emergencies. I think the COVID-19 pandemic was a very good example of the power of genomic surveillance and the importance of having open repositories. But I think we are way behind in plant pathology in applying these kind of approaches. So I'd like to argue that the traditional structures of science are not only too slow for emergencies, they actually hinder emergencies. We are just not set up to rapidly respond to these new problems. A list here of the reasons, some of the reasons perhaps, but I think we all know them. Funding, collaboration, open science is really important. Publication, I gave you the example of that Zambian outbreak and how it took forever to report that information. Even preprints were not used, and preprints really accelerate the dissemination and the sharing of new knowledge. You don't need to wait for peer review. You can have it peer reviewed afterwards. And the final point, I think, is something that I think everyone should also think about, is the way we assess science. Research assessment is generally hindering these approaches of open science and collaborative science. So we need to be more open also to the various contributions that scientists make and not obsessed with metrics. So let me very briefly again take you through some of these things. Just really want, I'm sure many of you are aware of preprints and bioarchive, but I just want to really make the point that this is the main point of preprints really, is to accelerate the dissemination of science. If that Zambian paper was posted a year earlier, we would have been able to contribute our bit a year earlier. So all of that got delayed by 1 year. And now there are news that the disease is moving out of Zambia to neighboring countries. So it's, you know, in the case of emergencies, time is of the essence. And I think we saw that, we know that I think how popular bioarchive became. Certainly in my lab now, we post every single paper in bioarchive without any hesitation. But I think the example of the COVID pandemic is also an interesting one, given that there was this surge in preprints. And pretty much if you're working on COVID or SARS-CoV-2, you'd be foolish not to post your papers as preprints, because there's probably more people doing this stuff. And also you want to really share the information, get feedback from the community and so on as soon as you can. But I also want to encourage your community to consider other forms of publications, these new forms of publications. So in addition to preprints, there are also mini papers. You can publish your posters, you can publish your data sets. You can have all of these formally published in platforms like Zenodo. This is the platform we really like. It's an open source platform that encourages the sharing of scientific information of all types. It's funded by the European Union, so I think it will be there forever. Stability is very important in the case of using platforms like that. But what's maybe more important than anything is when you publish in Zenodo, you get an author list and you get a DOI. So even if it's a poster, even if it's a video, even if it's a photo, you are actually getting credit for it in the proper way, which makes the unit you're publishing citable and also allows you to stake priority, so which is important for research assessment. So examples of these mini papers actually are the sort of the mini papers we published as part of that open wheat blast approach, where we encouraged everyone. We told the community that if you want to formally contribute, you have to publish your mini paper or your analysis or your data set in Zenodo. And then at the end, the current paper we are putting together on the African outbreak is actually a compilation of 6 mini papers that were published in Zenodo. So again, you can publish posters, data sets, genome announcements, whatever you like, and then you can cite them when you publish your formal paper, or you can incorporate them into a paper. So that's all I had, and I wish we can have a discussion about really rethinking our reward culture, because at the end of the day, we are the scientists, we tend to be the ones who also evaluate each other. And so it's the onus and the responsibilities on us first to make this happen. We really need a new research culture and a new reward culture to address plant health emergencies. So thank you for your attention. And I want to thank people in my lab, in particular, Torsten Langner and Jowin, who were the drivers there. Torsten is with the blue shirt and Jowin there is in the middle of the crowd with the red shirt. They were the drivers of the open wheat blast approach. And Adeline also right there in front, she was also very useful in terms of helping set up the biomaterial and arrange for the sequencing.
Thanks, Sophien, for that excellent presentation demonstrating how genomic surveillance using a community science approach can eventually lead to kind of information that can guide management practices, but also for the recommendations that you're making for making this more this citizen type of science more possible by changing the way we do things. So Sophien is connected with us, I've understood. So there's some time for some questions if anybody wants to ask. Yep.
Is there? Hi, hello. It is a very excellent and inspiring talk. In my case, for example, we face something similar with chili peppers here in Mexico, that we have to surveil water, air, soil, and the plant. And I think that this analysis can extend to surveillance, not just of the crop, like just in the point of the cultivation, but also seeds and water and any other environment that is involved in the crop management.
I don't know if you can give me some feedback about that idea.
Sorry, I have to say there was a lot of feedback.
Okay.
Hi, everybody. There was a lot of feedback. I'm not really sure if I understood the question correctly, but yes, clearly, I mean, this applies to all areas of science. I mean, not just plant health emergencies, obviously. But I think in the case of emergencies, in the case of infectious disease outbreaks, then it's even more important to be open and to be rapid and to quickly disseminate and share information, and also to bring together the experts who are actually the best equipped to address the problem. And so, I mean, yes, fundamentally, I agree. I mean, this applies to all areas of science. I think we're all hoping that, that we can communicate science in general earlier and faster. And I think that's really the whole philosophy of preprinting and Zenodo and all these various forms of pre-publication. But I think it's even more urgent in the case of emergencies and infectious disease outbreaks. I'm not sure if I answered the question because I didn't fully hear it, so I'm kind of guessing.
Yeah. Oh, can you hear me now?
I can hear you, but there's feedback, so I don't know if there's a trick to do on your side.
Can you hear me now without the feedback?
This is perfect. Yes, now I can hear you.
Okay. Well, I mean, it was just more a comment that we should extend this not only to tackle the emergency, but to surveil, for example, seeds, water, and soil, just to prevent instead of remedy this situation.
I see. I missed that. I mean, yes, absolutely. I think, for example, the reason why we invested time and energy in the multiplex amplicon sequencing is because that's like a dirt cheap technology that can be really scaled up to, for example, test seed lots that are being basically moving around and used in trade or being exported, imported. So you could easily imagine, let's take the case of wheat, that you have a multiplex amplicon sequencing platform to basically detect all the major pathogens and randomly test seed lots. And that would really be technically fairly easy and also not that expensive. I think a second point is I know also from my own experience that many countries have invested in actually PCR technology, have the know-how and also the equipment to do PCR because of the pandemic, because of the COVID pandemic. So now that we're phasing out of that pandemic, perhaps this is an opportunity to pivot to agricultural issues and plant health issues. And that would be amazing if some of that know-how and some of that technology would be actually now applied to pathogen detection. If we could sort of scale up surveillance and genomic surveillance, even if it's at the PCR level, that would be really amazing. I think actually there's opportunities in Latin America, and I've been talking to people in other parts of the world of even sort of more entrepreneurial initiatives where maybe some startup could be in place, be set up to really help establish some surveillance, some pathogen surveillance platform based on genomics in the broader sense when I say genomics.
Thank you. Any other questions? Marco.
Thank you, Sophien. I wonder if you have had any contact with other larger projects about this this idea of genomic surveillance or of sequencing large amounts of large number of species of plant pathogens, trying to collaborate with larger projects like the Earth Bionic Genome Project, where maybe we can propose them to have like a branch or something like that of plant pathogens being sequenced by these large proposals. What do you think?
Yeah, no, that's I mean that's a great idea. I've been involved primarily in the blast system and with some of the wheat pathogens, and I think we're now really considering this much more seriously because of the current situation in the Ukraine, actually, and between Russia and Ukraine. As you know, Ukraine and Russia are major exporters of wheat. I don't know if you saw Alison Bentley's, I think you heard her speak earlier today, but you can see her column that just came out, was it a couple of days ago in Nature, about the impact that this armed conflict will have on shaking the wheat trade. So I think it's actually very urgent now for many countries to step up and really establish some kind of genomic surveillance. I'm very happy to have discussion about that. With other crops, other diseases, I think it's also feasible. I know that there's a group, I think some of you here are perhaps involved in that. I know Jan Kurz is involved with SIP in terms of establishing a Phytophthora infestans, the potato blight, some type of genotyping platform. Again, I think especially with multiplex sequencing, it's so easy to scale up, and I'm happy to just let's get in touch and follow up. I'm happy to help make it happen, even if I'm not directly involved.
I actually have a question, Sophie. So one of the problems I've been facing with some countries is the ability to share biological samples and the legal issues around that. And I don't know if you've experienced that also, and how do you deal with that, especially in emergencies, right? When you need to get the material quickly.
Yes, I mean, this is always an issue, of course, it's clearly complication. So let me start first by saying that at the next International Congress of Plant Pathology, which will be in Lyon in 2013, if I'm correct, yes, it's 2023, sorry, not 2030. There will be a proposal to have a code of oath or was it code of conduct or for plant pathologists to deal with plant health emergencies. And it sort of covers some of the things I talked about there. Really, for example, that there is a responsibility to alert the community if there is a new disease being detected and so on. So it's something that's out there online. You can check the ICPP. If you can find it, let me know. But I hope this will be endorsed by the International Society of Plant Pathology in 2023. I think that would be a huge step forward. Now, the question that always comes up when we discuss the oath of the plant pathologist oath or the code of conduct is always that question. I mean, there's obviously very complex laws about sharing material. Even sometimes some countries put a gag on their scientists in terms of even informing the international community or even publishing about new outbreaks because of interference with trade and so on. So, I mean, obviously having a code of conduct is not about calling people to break the law, right? So obviously everything has to be done within a legal framework. But what really saddens me is so often the scientists themselves are actually the problem. It's not the governments. And that's really what saddens me more than anything, is the sort of this kind of perverse incentives we have in science. It's just, so that's why I really ended my talk by talking about how we need to revise our incentives and revise the way we reward scientists and the way we evaluate them. And this is a collective. I think let's start there. Let's not be too concerned about the constraint caused by government and by legal frameworks and all of that. So that's kind of how I approach that problem. Otherwise, I mean, I don't know if I want to open up a discussion here about how to sort of transfer materials and all of that, but there are certainly for example, if you work with the service providers like floodlight genomics, you're less constrained because that's if you want a tip. I guess I'm live now, so I shouldn't be talking a lot about these things, but there are ways, and I'm sure many of you know that there are ways to sort of get data out there by working with local scientists and by having local scientists sort of access directly the data. So I feel a lot of time, my job actually is to be a little bit a broker actually in terms of, a little bit like what Cabana does, right? In terms of sort of enabling local scientists to access technology and helping sort of alert them to sort of the opportunities they can access in terms of analyzing their data. And because everything is open and everything is transparent, then there's really no, it's very clear where the, who gets credit and who is contributing and all of that. It's all very open and transparent. Sorry, it's a bit of a rambling answer, but I hope at least I addressed part of your question.
So I think it's a very complex topic, but I think you answered it quite nicely. We're running well behind time, so unless there's one last burning question, I think there's one burning.
Thank you. Hi, Sophia. Well, always very nice to listen to your science and your opinions.
Good. Who are you? I can see you. So tell me.
Just wondering, how can we approach the funding agencies and governments to also to change the chip in this kind of how we evaluate scientists and over metrics?
Yeah, I mean that's I think step by step we have to keep on working on that and educating and alerting, just sort of I think going beyond simple metrics and all of that. Actually, Kath asked a question on the chat which is maybe relevant. I mean, let me just quickly read it. What would be your message to funders and how they might work together to help rapid response to pandemics and all of that? And my answer was that I'm a big proponent of Plan U. So I don't know if you heard of Plan U. Plan U is for universal. And I think Plan U is actually where the funding agency mandates the preprinting. So if you get a grant from a funding agency, you have to preprint all your papers coming out of that research. Or for example, the government, I don't understand why, for example, research foundations are not mandating preprints. I really don't understand. I live here in the UK and I don't understand why UKRI spends £22 million a year, 22 million pounds a year on publishing, on scientific publishing. They give that to universities and they don't mandate preprinting. For me, this is nonsense. I just don't get it. So can you imagine what you can do with 22 million pounds? I mean, that's actually 3 Sainsbury labs. So where I work, our budget, the Sainsbury lab, 100 scientists, we are 7-8 million per year. So so that's insane. And why they don't mandate preprinting, I don't understand it. So to me, actually Plan U, which was proposed by Richard Seaver and other colleagues who are leading the preprint movement, Plan U would be a great step forward because that will actually already open up science to everybody freely because everything will be on immediately posted, and then we can get organized from there. I think we can then it will be a huge push in terms of rethinking how we do everything. So yeah, anyways, it's not a simple thing, but let's be patient and we'll we'll get it done. I think it's certainly, I can tell you, I mean, I've been in this business now for quite a few years, and I can tell you that things are much better than they were 10 years ago, and certainly much better than when I was a PhD student, where actually publishing options were very limited. You could actually have a research project that you will not be able to publish because somebody has published something similar. So now we are in a very different world. So let's make the best out of it.